of Crop Science and Horticulture, Professor Villa, Deputy Vice Chancellor at Remix, Principals, Deans, Directors, Heads of Department, both academic and administrative, SUA, academic and administrative staff, SUA students, both undergraduate and postgraduates, if they are here, invited members of the public, and ladies and gentlemen, good afternoon. On behalf of the SUA community, let me thank you all for setting aside your precious time to come and listen to Professor Mwatawala, who is going to give us or to deliver his inaugural professorial lecture titled Facilitating International Architectural Trade Through Science, the Case of Tephritid Flies. According to the Oxford Dictionary, an inaugural speech is the first speech one gives when starting a new job. In our case here at the university, one does not give inaugural lecture every time one is promoted to a new rank. Rather, one is given the privilege of giving an inaugural lecture when he's promoted from associate professor to a full professor, which, as you all know, is the highest and the last rank in academic ranks. So definitely not everybody is given the chance to give these inaugural lectures until, according to our system, one reaches a full professor. So what do we expect to hear from these professorial lectures? From these lectures which are given by these professors, we are supposed to hear, or they are supposed to inform us and update us of the current and future research activities and directions in which they intend to go in the near future. They are also supposed to provide with an information on the opportunity, and also to give, they are given the opportunity also to inform us and also share with us and the general public what is being done and also the impact of their researches to the community. So definitely we expected to hear from Professor Mwatawala is what has been doing for the past years, he will tell us, and also the impact of this research to the community. Delivery of these professorial lectures goes back almost 40 years ago, when the then Faculty of Agriculture, Forest and Veterinary Science was part of the University of Dar es Salaam. And to all that, and I'm still to be corrected, as far as I remember, the first inaugural lecture, professorial lecture, was delivered by Professor Tana in the late 70s or early 80s. And this professor, I think he was in the division of veterinary science, I'm not very sure. In the 1980s, there were a series of these lectures, but with time, they've been decreasing in numbers. And as you all know, the most current one was by Professor Makundi, who delivered his inaugural professorial lecture 9 years ago. So this is why I spent some time to introduce to you, because this appears to be something new. So as the chairman of this meeting, my duty here is to remind, is not to remind the professors to deliver their professorial lectures including myself, I've never delivered one. Rather, my duty here is to chair this professorial lecture, which is going to be delivered by Professor Mwatawala. And the question of reminding the concerned professors, including myself, that I leave to the appropriate office. So, ladies and gentlemen, as the program shows, I was supposed to speak for 5 minutes, just to introduce or to give opening remarks, something which I think I've done. And the next event, which appears in the program, is to introduce Professor Maulid Mwatawala, and that will be done by Professor Gila, the Deputy Vice Chancellor Academics, who will take a maximum again of 5 minutes. May I now invite Professor Gila, who is also academic staff number 1, and also a professor who is due to give a professorial lecture one day. And I think Professor Mwatawala will take almost 60 minutes before we embark on the questions. So, Professor, in order to save time, I will request you to invite Professor Mwatawala to the podium when you are through with the introduction. Thank you very much for listening.
The chairman of this session, Professor Abeli, the presenter of the lecture today, Professor Mwatawala, the audience. I've seen some few students who have joined us. Thank you very much, ladies and gentlemen. I can't explain how I'm happy, because I was worried with the turnout today. We tried our level best to publicize this inaugural lecture. At the beginning, we thought we could go to multi-purpose hall, lecture theater 1, and I think we made a good decision to come here at IC. So we thank you very much for coming and for allocating time to this very important event. As you all know, delivering Professorial Inaugural Lecture is a requirement for every member of academic staff at the rank of professor. Although the practice is for all professors to deliver this lecture, if you go to the history of it, as narrated very well by Professor Abel, the chairman, it is basically meant for newly appointed professors. So I'm not sure if I'm newly appointed. It is indeed a rare opportunity to showcase and summarize one's research career and acknowledge the works of others, close colleagues and mentors. It is a way of getting people to know what academic or research activities a professor has carried out over years and what his or her future research interests are. Delivery of Professorial Inaugural Lecture has not been very smooth here at SUA, and the reason has always been, among others, shortage of funds to facilitate the preparations, which indeed is one of a big challenge. The last Professorial Inaugural Lecture was delivered by my friend there, Professor Rogers Makundi, on 25th November 2009. And I think if you count the years, it's almost 9 years. Professor Mwatawala, we thank you very much for coming forward to deliver this lecture today. We have on pipeline other two. For those who are not aware, Professor Mwatawala was promoted to the rank of professor last year, and therefore coming forward with the intention to deliver Professorial Inaugural Lecture is quite a good timing, and is in line with the definition of delivering this Professorial Inaugural lecture. So we thank you very much, Professor Mwatawala. But I'm happy to inform you that we have already 2 professors on the pipeline who have came forward after being sensitized by Mwatawala, and will be coming forward to deliver their lecture very soon. We will be announcing. Professor Mwatawala was born on 1st November 1970. He received secondary education from Sango and Bazengo Secondary School of Mbeya and Dodoma respectively. He completed Bachelor of Science degree in 1995 at SUA. He later obtained the Master of Science in Agriculture in 1998, and the PhD in 2008, all from Sokoine University of Agriculture. So he's a breed of Sokoine University of Agriculture. Professor Mwatawala obtained degree of Bachelor of Laws. On top of those 3 degrees, also he owns a Bachelor of Law from Open University of Tanzania, which he got in 2013, and the Masters of Law in Information Technology and Telecommunication, jointly offered by OUT, Open University of Tanzania, and the United Kingdom Telecommunication Agency, UKTA, of 2015. Mwatawala started his professional career at SUA in December 2000 as an assistant lecturer in the Department of Crop Science and Horticulture. He then rose to the ranks of lecturer in 2005, senior lecturer 2008, associate professor 2011, and finally professor last year 2017. He is currently the head Department of Crop Science and Horticulture since August 2017. He teaches courses on applied entomology, organic farming, and agricultural legislation and reduction of post-harvest losses and wasted through good agricultural practices. Professor Mwatawala is a member of Governing Council of Tropical Pesticides Research Institute, TPRI, a member of Ministerial Advisory Board of the Tanzania Food and Drugs Agency, TFDA, chairman on the Plant Quarantine and Phytosanitary Subcommittee of the Ministry of Agriculture, Livestock and Fisheries. He is also a vice chairman of the Tanzania Entomology Association. Professor Mwatawala also served as editor-in-chief of Tanzania Journal of Agricultural Sciences, TAJAS, between 2012 and 2016. He has been involved in several projects on integrated pest management for fruit flies in Tanzania, increasing value of African mango and cashew nut, resolution of cryptic species complexes of Tephritid pests to overcome constraints using SIT applications and enhanced fruits preservation using nanotechnology. Professor Mwatawala has published 64 articles in peer-reviewed journals, 1 book, a chapter, and numerous conference papers and extension materials. And as you see with this volume of publication, I think he's the right person now to come forward and deliver this inaugural professorial lecture. In his academic and research careers, he has received a number of awards, including the Best Student Award by the Ecological Society of East Africa in the year 2008. Professor Mwatawala's current research focuses on ecology and the management of fruit flies. And I think even the inaugural lecture today stems around that area. This is the biography of Professor Mwatawala. Ladies and gentlemen, after this introduction, I think now you know very much Professor Mwatawala. With this after this very short introduction, I now humbly welcome you, Professor Mwatawala, to come forward and proceed with delivering your lecture today. Please welcome.
Mr. Chairman, Professor Abel, Deputy Vice Chancellor Academic, Professor Gila, principals, deans and directors, colleague professors, members of academic staff, students, members of the press, distinguished invited guests, ladies and gentlemen. I'm greatly honored to stand here in front of you to deliver this professorial inaugural lecture. It will be about facilitating international agricultural trade, highlighting the role of scientific research, and with a number of cases highlighted from Sokoine University of Agriculture. Now, briefly, my journey through research started in a very odd way. So I started doing research on fruit flies out of an interest from another professor, Professor Robert Machangu, who is not around. And the story is Professor Machangu was very fond of guava, eating guava, the fruit. But he was always annoyed by the white maggots he would see inside the fruit. So he shared his experience to a Belgian colleague about his distaste for that. And he was linked to another Belgian expert on fruit flies based in Belgium, in Tervuren, Belgium. And through those talks, a research project was formulated, and it was hosted in the Department of Crop Science and Production, as it was known then. And the head of department then, Professor Amon Mairere, led the project and gave me the scholarship to do PhD under that project, and that's how I started. So it started with the guava fruit, otherwise it was not something that I knew I would be doing now. I would also like to say a little bit about myself, because I didn't want to come to, at first, my feeling was not about academics, but I have to thank someone, because my application for job at SUA was actually dictated by my brother. And he has traveled all the way from Dar es Salaam to here, maybe to prove that he was right. So I would like to thank my brother Shaban Mwatawala, architect, who is here with us today, because he's the one who dictated my application to SUA. So thank you very much. So I'm going to talk about the role of science in facilitating international agricultural trade. Okay, so food production is not uniform across the region of the world. And this is because of spatial and temporal variations in climate. So you have some regions with favorable climates for production of food, and some other areas that do not have that kind of favorable climate. But also, apart from spatial variation, temporal variation is also a must. So across the years, variations in climate affect the kind of production that we get from the fields. Now, in order to solve the problem of spatial variations in food production with the inherent spatial variation in food shortages, agricultural trade is very necessary. So food can move from areas of surplus to those regions of shortages through agricultural trade. So it's very important, and then it affects other things, including food and nutrition security of the nations, generates income for the farmers, and also helps to build economies of the countries. Now, unfortunately, this international movement of food, because normally agricultural products are moved in bulky quantities, and it's unfortunate that such movements also pose great risks of introduction, especially of pests, because various pests are associated with the commodity itself. So you can find a particular insect species living inside the commodity, whether it's a seed or it's a fruit or it's a leaf, depending. So as you move these food products, you also move, or there is a greater chance also of moving pests and introduce them to exotic places. Now, the International Plant Protection Convention is a UN treaty. So it's a multilateral treaty that was initiated by the UN FAO, And the major aim of the treaty is to facilitate international trade through coordinated action in order to prevent introduction of pests. But more importantly, IPPC aims at removing unjustifiable barriers to trade, because some countries may use an excuse that you have a particular species so we cannot trade with you, even though with some other motives, like political motives, and it has been happening a lot. So in order to prevent and controlling the introduction of spread of plant pests and products, IPPC was formulated by FAO. And trade is normally regulated by the WTO. So it's the World Trade Organization that regulates international trade, but WTO recognizes IPPC, especially the guidelines and the standards that are set by the IPPC Secretariat are the ones that are standard for setting plant health across the member nations of FAO. So that means if Tanzania want to formulate some phytosanitary laws and regulations, then it will follow the standards that have been set by IPPC through the application of sanitary and phytosanitary agreement. In other words, Tanzania cannot just arbitrarily set some rules that we don't want ABC without conforming to those international standards and guidelines which are set. Now, under IPPC, they have a set of what we call international standards for phytosanitary measures, which member countries can adopt while formulating their domestic laws on phytosanitary. So, a secretariat, as I said, at the IPPC is the one that is in charge of formulating the international standards of phytosanitary measures, and they provide to countries guidelines, recommendations, and expertise on dealing with quarantine pests, especially by providing measures to avoid unjustifiable barriers to trade. So there is an example of some two Southern African countries which were not in good political terms, and they used these as an excuse that we don't want your products because we have 1, 2, 3 pests, which actually the other country had. So they just used it as an excuse to bar trade from a hostile nation. So under ISPM, the international standards on phytosanitary measures, they define what we call a quarantine pest. That is an unwanted pest species. Now, by definition, the first thing, it has to be of economic importance. So it should be a species that cause a lot of losses, very high economic losses. So that is one qualification on one hand. On the other hand, if it causes such high economic losses, to qualify as a quarantine pest, then it should not be present in an area. So we can only call pest X is quarantine pest to Tanzania if Tanzania does not have pest X. So these are the two qualifications. First of all, it has to be of major economic importance, causing a lot of losses. And the other thing, it should not be present as yet, or present but being maintained officially. So that's the qualification. Now, coming to fruits. Fruits are important. I don't have to reiterate how important they are. We all know that fruits are important sources, is an important source of vitamins as well as minerals, etc. And the demand for fruits is on the increase globally because of changing lifestyle and demand for fresh tropical fruits and even temperate fruits. Now, with rising income and raising consumer interest, fruit trade, demand for fruit products is on the increase. But there is still a high potential for increase that is being constrained by biotic and abiotic stresses, as I'm showing here, including the quarantine pests that I just defined, that these are pests of economic importance, but to qualify as a quarantine pest for a particular region, they should not be present as yet. So, among them are this group of insects that we call fruit flies. And they're very important because they damage fruits by feeding on the fleshy part of the fruit. And normally what you see in a fruit are the white maggots or the larvae that you are seeing on the board. And these fruit flies are different from the fruit flies that we are used to, because we are very much used to fruit flies, the Drosophila, or we call them fruit flies of the genetics lab, because they are very popular in the studies on genetics. So these are different from those. These ones, we call them true fruit flies because they actually spend part of their life feeding and living inside the fruit. So they are true fruit flies. Drosophila is always attracted to rotting stuff. So these are the very small flies that you see in the kitchen if you leave a fruit rotting or a glass of juice that is decomposing. Then you have a lot of small black flies, and these are the Drosophila. So they do not attack a healthy fruit. While these ones, they attack healthy fruits, and that's why we call them true fruit flies. And one of the things that makes them of quarantine importance is because the major genera of fruit flies are limited to certain geographies. In other words, you have natives of fruit flies for each particular region. As outlined here, you have a genus, for example, called Anastropha, that you will only find in America. And then you have a genus called Raguletis, for example, that you'll find in Central America. And then you have the Bactrocera from Asia. And then you have the Ceratitis, which we have here in the Afro-tropical region. So their geographical distribution is limited. That means if you see a Bactrocera in Tanzania, it means it has invaded Tanzania from Asia. And if you see a Ceratitis in Europe, it means it was introduced from Africa to Europe, because originally their distribution is limited according to these geographies, and that's why it makes them of quarantine importance. So they cause heavy losses, as I said. As a result, they are regarded as a big threat to food and nutrition security of the various nations. And the losses is both in terms of quality, because the yield losses are very high right in the field, but also the qualitative losses are also very high because consumers are good, even if quantitatively high. So, and a brief on the life cycle, because in order to understand the kind of damage they cause and whatever that I'll be talking about, it's better to give a very brief description of the life cycle. And normally you have the adult fly laying eggs inside the fleshy part of the fruit. So these are different from the insect that you find inside a seed, for example, of the mango. These are found on the fleshy part of the fruit. And normally, it is the larvae that cause damage because they feed on the fruit and they actually live inside the fruit. So they cause damage because that feeding is accompanied by rotting. And then at one point, the larvae will pop out of the fruit and pupate in the soil. So they'll form a hard cover, the puparium, and bury themselves a few centimeters under the soil. They can either do that by popping out, or a fruit, because it has been damaged, it will drop, and then the larvae will emerge and form a puparium. And then finally, the adult will emerge and the life cycle will continue. And that's why we call them the true fruit flies. Now, our research at SUA started in 2014, in 2004. And as I said earlier, it was a main collaboration between Sokoine University of Agriculture and the Royal Museum for Central Africa, based in Tervuren, Belgium. And that photo was my first day in the field. So that's the first day I started to work on fruit flies. We were setting up the traps. And we had two phases of the project, the main phases, 2004 to 2008, and then a second phase from 2009 to 2013. So like 9 years of field research that generated whatever information that I'm going to present today. Now, previously, limited research on fruit flies was done in various parts of the country in uncoordinated fashion. But through this program, between SUA and RMCA, we had other smaller collaboration with some other partners that also contributed to a lot of information that I'm going to present today. So, when we say that we facilitated international agricultural trade, it's because we facilitated that trade through this process that is called pest risk analysis. Now, ideally, because we said we don't want to have unjustifiable barriers to trade. So, two countries, one may have a pest X, and another country may not have a pest X, but they want to trade. Mere presence of pest X should not be a barrier to another country from importing products from another country. But you have to do it with minimal risks. Now, in order to determine the kind of risks that you may face, you conduct what we call pest risk analysis. And through that process, you will be able to determine whether that pest X can establish into a country or not. So you do the PRI, so probably it's just a risk analysis, and in the end you'll be able to make a decision whether to import product from an infested country or not, depending on the outcome of the risk analysis. And what we're saying is, it's a biological process, as we're saying, so, sorry. And what we're doing with pest risk analysis is simply to evaluate scientific evidence. So it's like in the court of law, but this is different kind of evidence. It's scientific, biological evidence. And through that process, we will be able in the end to say whether or not, no, we should regulate, we should control this pest using laws, by laws saying that we don't want it and it's mandatory. And if so, how strong should those barriers be? Should we say that we don't want your products altogether, or we want your product after treating with products 1, 2, 3, or we want your product after even minor field treatments, then you can sell it to us. So that's how we facilitated. So we provided, as research, we provided biological scientific evidence that was used in so many different cases as the main input through the pest risk analysis. So the major aim, as I said, is to minimize introduction of pests. A good example, most of us must be familiar with the introduction of the larger grain borer, which we call Scania or Dumuzi. If you want to feel the consequences of not having done properly the pest risk analysis, you end up getting such introductions that you may not be able to control in the future, and your production is under jeopardy. So what we do is to minimize the introduction of pests through movement of fruits, but plus other agricultural products. And when you do pest risk analysis, the first thing you have to do, because we said with quarantine pests, the first thing you have to know that it is of economic importance. Otherwise, it's not a quarantine pest. It's just another pest. But the other thing is it present? So these are the two important things. So if Tanzania want to do business with another country, the first thing we'll ask, what kind of pests do they have? And we'll ask ourselves if we have them. If we do not have them, then those pests become quarantine pests to us. And if we want to do business with them, we have to do pest risk analysis to minimize chances of introducing pests from that country. So identity and presence of a pest in a particular area are the very first stages of conducting a PRI. Because if something is not of economic importance, and if it is already present, then it's not a quarantine of quarantine importance. So that's the basic. Now, so among the things that we did, or that came out of all the research that we did, can be grouped into major 4. And the first one is on detection of invasions, and I'll explain that one. And the second one was resolving the identity of some species, what we call cryptic species, which I'll explain later on, but also description of new species. So these are species that were not known before, so they are new to science. But also on providing information that would assess probability of entry, establishment, and spread of pests. So in that case, you may trade with one country, and the pest may come with the commodity, but will it be able to establish itself and later on spread? So all this information we provided. And so I'll start with the role of science in what we did to facilitate those stages. And I'll start with what we did about identity and presence of pests in order to facilitate the PRI process. So we went as far as borders of Burundi. Sometimes we're dripping wet in the rain, but that's what you have to do in order to detect these invasions. So what we are saying is, I said that these fruit flies most of the pest genera are of limited geographical distribution, but some of them have moved to areas beyond their origins. So a good example is the fly that you see here on the board. This one is called the Mediterranean fruit fly, that is of African origin, but now it has spread to almost every part of the world through trade. So it's Tanzanian, I mean not Tanzanian, Afrotropical origin, but now spread to so many various parts. So they've been intentionally or accidentally introduced, but such introductions are a big threat to fruit trade. So a few cases now of what we did. So there is this case of what we call Bactrocera invadens, which originally was given a common name of the African invader fly. In 2003, we sampled mango from Matombo area, and we got some flies out of them. And the reason for sampling was to take those specimens for my training on taxonomy of fruit flies. So I need a specimen. Now, out of those specimens, about 4 individuals were very different from the natives. Remember, I said they have limited origin. So we can tell that this one is Asian, and this one is African, and this one is American. So 4 flies out of them were quite different from the natives of this region, and we suspected an introduction. And these samples were later taken to experts on taxonomy in order to confirm. And it was confirmed that this species has just been detected in Kenya. So a few months ago, the same species was detected in Kenya. And by then, it was thought to be new to science. So it had never been described before by then. So we had the first Kenyan detection, and then the second Tanzania detection of a species that breeds in high numbers, which is supposed to be very new to science. So everyone was frustrated, and the losses that were caused by this species were very high. So that was the first case where we detected invasions. And we informed the world that we have this kind of invasion so that these countries could protect their industry. Here we are talking about the global food security vis-a-vis production in a country. A country may lose markets, yes, but the global food security will be protected. So there is a second case here of invasions, and this is another fruit fly species, also of Asian origin, which we call Bactrocera latifrons, or the Solanum Fruit Fly. Now, this one was detected right in the horticulture unit at SUA. So we had several samples of flies, and suddenly we saw one fly that was slightly different on color patterns. When we identify these flies, we look at color. They are very colorful flies. Actually, they're very beautiful flies, apart from the damage that they cause. So they're very colorful, and we use colors to identify them. So when we see a different color pattern, we suspect things might not be the same. So we detected this one also in 2006, and then we sent specimen once again because I have to get confirmation from taxonomists, and it was confirmed that it was Bactrocera latifrons. And this was the first detection in Africa. So maybe it arrived in Africa a long time ago, but there are no research activities on detection and surveillance. So this was the first record of Bactrocera latifrons in Africa, which we detected at SUA, and later on it was also detected in Kenya a few months later. And this one was also reported. And then, so those two were the detections that these species came from abroad and invaded us in Africa. If I go back to the case of Bactrocera invadens, the one which I said was thought to be new to science. And then there are 2 schools of thought. Some people say, no, this is not a new species. It is similar to another species in Asia that we call Bactrocera dorsalis. So there was no point in giving it a new name of invadens. And now this was taxonomists arguing. And another group said, no, this is a species in its own right. It deserves a new name, and it's quite different from dorsalis. And a good question people ask is always, what's in a name? So what's in a name, of course, is nothing, except if you're a fruit fly. If your name changes, your identity and everything also changes. So names matters a lot to this species. So what's happened was the International Atomic Energy Agency, because they're the ones who facilitate the trade through application of SAT. So we formed a team, an international team, to resolve this plus other species, the identity of this and other species. And we did not confine ourselves to the definition of a biological species, the one that we commonly know. Rather, we sought some other biological evidences to confirm whether this is the same or different species. And if you look at the list, so we looked at molecular, cytogenetics, pheromones, behavior, and all these other biological entities in order to say whether this is the same or different species. And the evidence came out that this is just the same species. So all of a sudden, the name Bactrocera invadens disappeared. So we may still have posters all over here, even in our department, with the name Bactrocera invadens, but Bactrocera invadens is gone, and now what we have is Bactrocera dorsalis. Now, in a way that becomes a relief to us, because those people in Asia cannot quarantine our products now, because Bactrocera dorsalis is present, remember from the definition, because it's already present, it cannot be a quarantine pest to them. So we can also sell our products to Hawaii, for example, because Bactrocera dorsalis also invaded Hawaii, etc., and so many other places. So the taxonomy of B. invadens was rejected, and it became Bactrocera dorsalis. So, and you can see this was a very big team, because there was a lot of arguments, and so many experts were needed. So I got 0.000 something points maybe from this one. And these are the people. So this was our first meeting in Vienna, in the headquarters of IAEA in Vienna. It's very easy to pick me out. Then there is a case of another, this one is native. It's a native species. It's called Cerathitis rosa, or the Natal fruit fly. It's also a very important fruit fly species, attacking fruits. An interesting thing about the Natal fruit fly was when we were sampling here at Morogoro, remember we did research from 2004 to 2013, so we have a lot of trapping data for all those years. An interesting thing was that the variations, if we sample from here all the way up to the Uluguru mountain, some 1,600 meters above the sea level, the populations were more or less the same. And it couldn't be the case, because with these changes in weather and altitude, we expected some major variations. But also, temporarily, I mean, if you sample now when it is very hot, and when you sample in June, July, when it is very cold, we used to find substantial number of Cerathitis rosa. So it was a bit of a puzzle that how can a species adapt itself to so much wide variations in climate. But we were not the ones who were only puzzled. We had some people in South Africa who did some research and concluded that Cerathitis rosa is adapted to warmer climates. And of interest now would be what kind of restrictions Europe, for example, is a temperate country, would have on fruits that are attacked by Cerathitis rosa, because it prefers warmer climates, and Europe is cold. It means the risks of introductions are very low. So they're protecting the trade that way. But some other people in Reunion came up with different results, and they said Cerathitis rosa is more adapted to colder climates, which means potentially it can establish itself in Europe, which means now the quarantine measures against products that are attacked by rosa would be very strong. So we had these arguments. Anyway, so through research and through the evidence on the various parts, molecular biology, morphometrics, developmental biology, cuticular hydrocarbons, etc., it was resolved that we were dealing with two different species under one name of Cerathitis rosa. So we had one species that is adapted to colder climates, places like in Nyandira, in the Uluguru mountain, and we had another species that is adapted to warmer climates, the areas like here we are like 500 meters above the sea level. So, in the end, this species was now described as Ceratitis kilisi by Dimea, Mwatawala, and Virgilio. And the man you see, the first one on the left, is the late Serge Kilisi. Some people in here know him. He was a very good researcher on fruit flies based in Reunion, working for CIRAD. So he was a French national. And apparently, Professor Marel will correct me, he's one of the people who came here to assess the status of fruit flies way back when we had the Franco-Tanzania project on horticulture. And that is Dimea. So it's Ceratitis kilisi, Dimea, Mwatawala, and Virgilio. So it is a new species, new to science. It never happened before. And then there is a case of another fly, a native fly that we call Ceratitis cosaira. It's a marula fruit fly, or a mango fruit fly, because it attacks marula and mango. Now, the curiosity that always killed the cat. We were curious about the identity of this fly as well, because we were trapping some flies that were very, very small in morphology. Ceratitis rosa is relatively large compared to whatever that we are getting from a particular trap, because we collect them by trapping. So these flies were very, very small. But if you look at them morphologically, they resembled Ceratitis cosaira, the marula fruit fly. And we suspected a different, but this one was easy to resolve because just morphologically, it could be resolved that we were also dealing with two different species, the proper, or the Ceratitis rosa, cosaira, in strict sense, which was larger in size. And then there was another one, which we came to describe as Ceratitis pallidula, Dimea, Mwatawala, and Virgilio, which is this one. And then there are some more evidence from DNA, barcoding, etc., which resolved that these two were different species. So, out of that, we could resolve identity of three species. The first one was Bactrocera invaders dorsalis, when we said they were all dorsalis, one and the same. Then we had Ceratitis rosa, the natal fruit fly, that we are dealing with two different species, Rosa sensu strictu, and then Ceratitis kilisi, Dimea, Virgilio, and Mwatawala, and Virgilio. And finally, Ceratitis cosaira, which were also two different species, cosaira in strict sense, and Ceratitis pallidula, Dimea, Mwatawala, and Virgilio. So these ones could be resolved. And the implication is the same, that these species might or could have gone undetected into other countries, misidentified either as cosaira or as rosa, and left to breed. And later on, when the populations are very high, the losses are very high, and they cannot be controlled, then you realize that you have an introduction or an invasion. And then this one. Now, out of this work that we are doing, we found some other species, not related directly to fruit flies, but they are under the fruit flies project. And we have a taxonomist who worked here. He was working on moth, not flies now. But he found several moth species, and he was happy to name one in my name, and that is Phalonoricta Mwatawala, which if you Google from Wikipedia, that's what you'll find out. But fortunately, it's not a pest, as yet. So that was on resolution of cryptic species and identification of species and detection of invasions. Now, the other thing that we have to look at in order to determine whether or not a pest can introduce itself and establish itself and spread is to check on availability of preferred hosts, as well as weather condition, and whether the new introduced pest can actually compete with the native ones, because otherwise it may not be able to establish and spread. Now, we did a lot of sampling also on the fruits, but the first one is just a general scientific explanation on how do fruit fly choose hosts. Because we're talking about preferred hosts, they don't just oviposit or lay eggs on the hosts. And one of the things that is known and in theory that the mother of the fruit fly is the one who knows best about what the kid should have, because they have no choice. The egg has no choice of where to be laid, but the mother chooses the host in a theory that is popularly known as mother knows best. So we sampled quite a lot of fruits, And have in mind, back then we thought that Bactroceras invadens was new to science. So whatever we find out from the field was new to science. So that was the advantage back then. So if I see that tropical almond is affected, then it's a new host because no one studied the host before. Or if marula is affected, then it's a new host. Or if lucky nut, Tevetia peruviana is affected, it's also a new host. So that was very easy part of the research. Whatever you get is new. So we sampled quite a number of new hosts, but the most important thing was to get just a general host range of fruit flies in Tanzania because we were not dealing with Dorsalis only. We were dealing with the biodiversity, the whole biodiversity of fruit flies. And we sampled that from Morogoro only, 35 fruit species are attacked by fruit flies. So that is a very high number. So almost all fruits that you can think of that you find in the lowland is basically attacked by fruit flies and it's a potential host for fruit flies. And the one that was introduced by Professor Dorsalis had a highest host range. We got it from 27 fruit species. That include most of the commercially important fruits, avocado, mango, guava, papaya, even banana. So it's a very important pest, and that one was was reported. But also remember we had latifrons was new also in our area, not new to science, but it was new to have new hosts. The hosts that we have are different from the hosts we have in Asia. So we also sampled a lot of hosts. Now, fortunately, this latifrons is limited in terms of family that they attack. So this species attack only Solanaceae and Cucurbitaceae. So tomato, watermelon, and stuff like that. So we sampled a lot, and most of these were new hosts, I mean, probably because they are not grown in Asia. So we had the African eggplants, watermelon, and so many other hosts. And these were very important because they are normally the reservoir hosts for the pests, some of these. And remember, I also said that we have to check whether the climatic conditions are also suitable for establishment of the pests. Now, what we normally observe from the field are the actual numbers, whether we trap them or even seeing us in this room is just the actual numbers. But we don't see the underlying biological processes that resulted into this population here. So it's the same with the flies. So there are so many underlying factors behind what we observe in the field, including the basic population processes. How many migrate into an area, how many new offsprings are born, how many die, how many move out of an area, etc. As well as how climate affects the population. Human activities, we spray herbicides, we practice different types of cultural practices like crop rotation. All these impact the kind of population that we observe, the ultimate population that we see. We also have competition. Organisms always compete, and the one that outcompete the other will survive. The one that is outcompeted will perish, and that will also have effect on the population. So this is just the background on the underlying factors. Now, because we sampled for so many years, I mean, like I said, from 2004 all the way to 2011, the data that are presented here, and we could get these peaks. It's not easy to get an image with very good resolution because of the length of the y-axis, the x-axis. But it showed that from 2005 all the way to 2011, we collected these flies, and the peaks kept on repeating themselves. So we had the same temporal pattern over time. Now, if we go to the averages, now with this one it's easy to see because now we average the populations for January all the way to December across all those years that we sampled, and the pattern is very clear because you can have very high populations from January all the way to June, and then very low populations all the way to October, because it tells you that this species prefers warmer, humid climates, different from cold, dry climates. So you could get that pattern out of all those years that we sampled. Now, it tells you a lot because importing countries now can pick which window of the year to trade with you in order to minimize the risk. If they trade with you during February, March, then the chances that you are going to get the pest into your country are very high, compared to chances if they trade with you in August and September when the populations are very low, then the risks are very low as well. So that was the temporal balance. But then we're involved in determining now the global distribution of this species by then it was known Invaders, as I said. But before we could publish this paper, it changed its name. So the name changed to Dosalis along the way. But when we started, it was like we were dealing with invaders. So it was a regional collaboration, and we had sampled, we had placed traps in 6 locations in Tanzania: Makutupora, Mlingano, Koroge, etc., in Oldeani, in Arusha. And for 2 years of this species in those locations. And then we used a model called the Climax in order to predict the chances of this species in establishing itself in other regions of the world. So we have a whole map of the world, but what I'm showing here is just a part of Africa. Now the red part is the part that is susceptible, or was susceptible, to introduction of Bactrocera dorsalis. But what we are saying is all parts of Latin America, southern parts of America, the Mediterranean, and the tropical Africa for introduction of Bactrocera dorsalis. And actually now it has spread throughout the tropical parts of Africa. And then came the case of Bactrocera latifrons, which also invaded us. We also had to do some spatial distribution, studies on spatial distribution, but this one was very limited. We moved along all the main routes to the borders with Rwanda, with Uganda, with Malawi, Zambia, all the borders coming to us. So Horohoro, Holili, Taveta, we moved along the routes leading to those borders and placed the traps all along. But luckily this one was not very much distributed, so we could find it from Morogoro, coastal, parts of Tanga, Kilimanjaro, and then there was one sample from Mwanza, which we don't know how we got it, but at least it was there. But most parts of Tanzania did not have Bactrocera latifrons, and the implication would be if you want to produce, if it's a quarantine pest and to produce for exports, we can actually avoid those areas and produce in areas where Bactrocera latifrons is not present. So that was about the climate and the host. But the other thing that I said is they must compete. So they can be introduced. Just like us human beings, we have to compete. Insects also have to compete. Otherwise, they'll be extinct. Now, when a new species come, which means that is an invasion, that's what we call an invasion. When a new species come in, it must compete with the native species. Now, 2 scenarios can come out of that competition. Either they can end up coexisting, so it means the introduced and the native species will coexist together, or it may happen that one species may displace the other completely, so the other one will be thrown out from the niche and dominated by the other species. So those scenarios can come. Now, in these studies, normally we say species that compete, they compete very well, have certain traits, while species that invade very well also have some different traits. And I look at this scenario where you have competition. If you are able to survive in competition, it means the areas that are habited by these pests, first of all, are very favorable. There are so many species, crowded, the resource is limited. The outcome would be even the reproduction rate of those species will be very low because you don't reproduce too much if you are limited in terms of resources. It's just like family planning that we're doing. So insects do that as well. If they don't have enough spaces, not enough food, then they lay very few eggs. So that is a strategy to live in a crowded, competitive environment. If the room is open, there is a lot of space, plenty of food, then they reproduce in very high numbers, no problem, because there is a lot of food and a lot of resources. So, and then you have the competitors. The competitors do not move, they don't run away, they stick around and compete. So the population is always more or less stable, like the species that I'm showing below. So the hypothesis was that one was a very good invader and the other one was a very good competitor. Now, the question we asked was, can it be both, that a good invader and a good competitor? And we got the answers from studies on biology. where we had these two species. One we thought, the top one we thought is a very good competitor, and the other one we thought is a very good invader. And when we compared them, so one is a native species and another one is an introduced species. When we compared them, we found that the introduced one had better traits than the native one, because first of all, it can grow very fast, it lives much longer, it lays much higher number of eggs, the population doubles within a very short time, and they produce more eggs. Now, those are very good traits for competition, because if you produce more eggs, you live much longer, your population doubles within a short time. That is biological success, because biological success is measured in numbers. So the higher the numbers, the higher the success. So based on that, it means this species, yes, invaded us, but also outcompeted our native species. So it had both traits of competition and of invasion. Now, finally, the other thing is, if you want to do business, then the other thing they'll ask, maybe, yes, this part is introduced, is infested by particular fruit flies, but are there some methods of controlling it so that the risk can be mitigated? So, we did several research on integrated pest management of fruit flies, but generally, the most accepted model is that the control, I know, because most of the question might come from this part of the lecture, which I will give it very limited time anyway. So it's as simple as 1, 2, 3, 4. Monitoring, sanitation, spot application of bait, and male annihilation. Unfortunately, the second one, which is the major part of it, we don't like it. We don't want to keep our orchards sanitized. So we just want a single bullet, boom, and the flies are dead, and then that's fine. Now I can harvest my fruits. Now, along the same, we formulated a local bait. We didn't finish it. We formulated a local bait because the flies, they want protein, they want sugar. So these are the sources they want. And normally the females, they want protein because they want protein for eggs. So because of the protein that is used by eggs, they're always craving for protein, while the males, for some other reason, they need sugar. So if you mix protein and sugar and a toxicant, you can get a very good bait against fruit flies. So that's what we did, and we tested it. So these are some of the results where we tested the 3 IPM programs, one using the local bait, another one using a standard insecticide, another one using a standard bait. Now, what we found was, in terms of incidence, how many samples of fruits are infested, the botanical bait and the insecticide spray, the performance was more or less similar, except for the spinosad bait. So the insecticide, the standard one, performed in a similar fashion to botanical bait, and the same one for infestation. These are the two different indices that we use. The first one is simply how many samples out of the total are infested. So if you have 100 fruits, if 4 are infested, then the incidence is 4. The other one is how many flies will come out from a single kilogram of fruit. So you can have 1 fruit that will give you 10 flies. 1 fruit can give you 100 flies, and that is infestation. So these are the 2 different indices. And based on that also, all these standard programs using insecticide, standard insecticide products, a standard bait, and a locally formulated botanical bait, the effects on flies' incidence were also not significantly different. Then there came the weaver ants also on mitigation. So these ants are predators. They eat the other insects. That's what predation is. And when we did research on temporal nature, we found that weaver ants also perform very well against the fruit flies, although the mode of action is not very well known. But here we are comparing the performance of weaver ants to a no-control orchard, and an orchard that was sprayed by an insecticide, imidacloprid. And you can see that the performance of weaver ants was very good. The only problem was we could not sustain very high populations of weaver ants that is needed for effective control. We need higher numbers of predators in the field. And these are the reasons, because poor rearing conditions, lack of foods, predators, etc. So we decided that we should generate a method of introducing weaver ants into the field in case the populations go down. So you go to the wild settings, look for the weaver ants and introduce them into an agricultural field. But the success of that depends on locating the queen of weaver ants, because these are social insects. It's only the queen that reproduce. The others are workers and pests. So when you go out in the field, you find so many trees. And these nests, they look the same. But thanks to our PhD students, we could develop a method and we can go out in the field. And even if we have 100, 200, 500 nests, we can go and point out that that nest has a maternal queen and we can harvest it. So that will sustain a colony, because otherwise if you harvest other nests without the queen, that colony will ultimately disappear. But if you have an egg-laying queen, that colony will keep on going. So we developed that method. And then normally weaver ants, the queen of weaver ants, the ones that have not been fertilized, spent a lot of time hunting for these natural flies in the field. And because there is another Asian species of weaver ants that fly, the natural flies takes place in the morning, we thought the same. So we used to wake up in the fields in Muheza and go to the field very early in the morning at dawn, waiting for them to fly, and they never flew anyway. So later on we thought that we should put a camera. So with the assistance of camera, we tried to set so many cameras in the field, then we could observe when do they fly. And they fly in the evening, not even in the morning, and they fly during the rainy season. So the implication was, when they fly, they mate and they fertilize the queen now, they return to the ground and start forming their own colony. Our objective was to collect the fertilized queen and put them in artificial rearing systems so that they reproduced much faster than in the wilderness. Now, out of all this, as I said, we generated a lot of information to facilitate trade through the PRI process, as I demonstrated, through detection, identification, etc. Now, in the end, what we did, or that came out from the project for the Tanzanian government, was development of this management plan for fruit fly, which is a very comprehensive plan outlining all the research priorities and what should be done in the future in the field of fruit flies. And then because of the expertise that has been gathered for so many years at SUA, it was regarded as a center, not of excellence, but of expertise by the other partners in Africa and Europe. And based on that, we host an international course on fruit fly taxonomy and ecology after every 2 years. And it attracts students, like 15 students from all over Africa. So it's an international program that we host, and we train students from, or candidates from many parts of Africa. Otherwise, I would like to thank the employer, the government of Tanzania, thank to the management of Sokoine University of Agriculture, Vice Chancellor, Professor Chibunda, Deputy Vice Chancellor, Professor Gila. Thank you so much. I would like to thank people who supported all these works, starting with the Belgian Development Cooperation, DEGOS, IAEA, International Foundation for Science, and DANIDA. Thank you so much. I would like to thank my former supervisor, or the people who supervised my master's and PhD studies, Professor Rhodes Makundi, Professor Amon Merere, and Dr. Mark Dimea, who's not here. I would like to thank the members of academic staff of Crop Science and Horticulture, Department of Crop Science and Horticulture, and thank to a long list of my students, PhD and masters, the supporting staff. Mr. Senkondo is not here, Resta Maganga, she's not here, maybe she's still around. The late Yasin Mgoba and John Kusolwa, they contributed a lot to what we did. And last but most, thanks to my family, and they're here with me today. And thank you so much.
Thank you very much, Professor Mwatawala. Members, I think you will agree with me that the lecture was very clear, and I'm afraid that we will have many questions because everybody now knows what is this fly. Before I invite the one to give a vote of thanks, let me recognize the presence of the Vice Chancellor who has just joined us, Professor Chibuda. You are welcome to the inaugural professorial lecture. So can I now invite Professor Makunde to give a vote of thanks on our behalf, please. Professor Makunde.
Honorable Chairman, Vice Chancellor, Professor Chibunda, Deputy Vice Chancellor, Academic Professor Peter Gila, Professor, Deputy Vice Chancellor, Administration and Finance, if he's around, in his space, college principals, directors and deans, professors, the whole academic staff, heads of academic and administrative units, students, myself, a number of them around, media, invited guests, and all other participants in the audience, ladies and gentlemen. First of all, on behalf of Sokoine University of Agriculture, and the entire team led by the Deputy Vice Chancellor Academic, who organized and arranged the presentation of Professorial Inaugural lecture by Professor Mwatawala. We wish to pay homage and say thanks to Professor Mwatawala for being our guest speaker today by delivering an inaugural professorial lecture on the facilitation of trade, international trade in agricultural produce by using science. I'm sure you have seen all the slides that have been presented. It's all science. There is no history, there is no anything else but science, pure science, but science. And therefore, Mr. Chairman and the audience, join me to clap 3 times very strongly for Professor Mwatawala, who has presented a very informative and interesting inaugural lecture.
Right.
Thank you very much, Professor Makunde. We had assigned 5 minutes, but it's taken almost 15 minutes. And this will limit our 40 minutes, which were been assigned for questions. But probably because he's your student, you don't want him to be asked questions. So many of them. But now, from 40 minutes, we have only 30 minutes for questions, which means that we shall take at maximum 7 to 8 questions. In other words, you spend 1 minute to ask your question, and I don't know how we should agree. Should he be responding to each question, or should we collect 3 or 4 questions and then we let him respond? Okay, so Professor, maybe you could be writing somewhere. So who's going to be the first to ask? As I said, spend
minimum time to ask, so that others can get the chance also to ask. Professor Ruben.
Can I have a mic? Based on this differential severity and incidence of fruit flies during the year, from January to December, you see that there is a time when we've got maximum levels and then minimum levels, and then with that kind of variation. Do you base solely due to weather differences, or it could be confounded with availability of fruits during those periods? Number two, did you take on board the biological species concept, which takes on board reproduction barrier in deciding whether the species are the same or different, or that concept is out of date, and then you took on board other procedures for deciding whether the species are the same or different? Thank you, Mr. Chair.
Thank you, Mr. Chairman. My name is Wahab Kimaro. First of all, I should commend the presentation which was made by Professor Mwatawala. I really learned a lot from him. My background is not crop science, but I captured a lot of things which are very beneficial for me as a stakeholder in agriculture. I would like to know the bait and the control mechanism he used. He tested pesticides, biologicals, bait, and weather ants. So I'm interested with these weather ants. I know the work of this weather ant was comparable to the use of this pesticide, but there is a challenge of working in the field with this weather ant. I don't know what is he advising farmers on choosing the control method between the pesticide and weather ant. Again, the second one, I'm interested to know the whereabouts Philonectar Mwatawala species. Thank you. Thank you. Any question from my lady? Could you tell us your name, please?
My name is Sokoine. I've got A, B, and C. The first question, I'm interested to know how your research findings have supported export trade in Tanzania, and what are the way forward? And then the second, for part B, is you have done a lot on the use of weather technology to control fruit flies, but I'm more interested in seeing how practical is that at farmers at the moment. And the lastly, the fruit fly is still a problem among small-scale farmers, despite all the knowledge you have developed on this. What should be done? Thank you very much.
So far we have 6 questions.
Thank you. As you are all aware that we were live on Facebook, and we have a question from Professor Gerald Isinzo. His remarks. A question from Professor Isinzo. Congratulations for this well-deserved achievement. If I had $1 billion for you, what would you do Pest.
I listened to everything, but when you mentioned about Marula, I got, I said, what is this? Now, Marula is Ngongobori. It's the one that is supposed to, where we get to Marula, and if you go there, you see a lot of Marula. It's Kelokalia Birera. Some years ago, the retired president declared the tree as the tree of the year. And they planted the tree at Chakanikemi at the University of Dar es Salaam. Now, we have a lot of Marula trees around in the savannas, and they have the potential to contribute to the livelihoods of the people. Now, those trees are very huge. Now, from your presentation where you say, strategies to control them, I'm just questioning how do you advise communities where these trees grow to actually use those techniques because they are so huge. And I'm just imagining how someone climb on those trees to put baits or spray them, because potentially it has something to do with the livelihoods of the people. Thank you.
Professor Kisepo, when I'm asking people to introduce themselves,
Okay.
for your own interest, was going to not too many people here know you.
Thank you for, okay, my name is Penyanguka Kisepa. Not because Marula makes Marula wine, but for sure, indigenous fruits, they are being promoted and they have a role to, I work in the Department of Food Technology, Nutrition and Consumer Services.
My name is Catherine Suyabengas. My question is, when we eat foods or animal food contacted by houseflies, one can suffer from cholera. So, by chance, if someone will eat their food contacted by these fruit flies, what will happen?
Two more questions, please, and then we'll give you a chance to respond.
Congratulations, my question is on, you've spoken of insects being a pest, but from my side as a nutritionist, I see a huge potential of using insects as a source of protein for fish. What is your opinion, and what do you think could be the most promising species of insects which you have studied over a period of time?
Congratulations, Professor Mwatawala. Coming from the wildlife department, I'm not an expert in culture, but based on your lecture and your animals, I find it difficult to distinguish or to separate the proliferation of your animals you've been talking today and the fruits we are finding in the market right now. There are quite many varieties. Each day in the morning you find another mango. So many varieties. Why don't we control these fruits first before controlling your animals? Because I find something wrong about it.
Ladies and gentlemen, I think we have received more than 10 questions from the floor. And let's give him a chance in the first round. And then if time allows, we shall go for the second round. So, will you respond?
Okay, thank you so much. So I'll start with the question from Professor Shazia Omkuku wa Mnyuku Ruben. So, so Professor Ruben asked whether when we study the spatial abundance of insects, did we take into consideration other factors apart from weather? And the answer is yes. That's why before introducing that part on spatial and the temporal variation, I explained the underlying causes to whatever we observe, and I gave an example that because some people migrate from sewer, some people come to sewer, some people die, some people get born. And it's the same for insects as well. So there were those, the biological processes, birth, death, migration, immigration. There was competition. I said you must be able to compete, otherwise your numbers will go down. You have predators that will attack you in the field. Then we talked about life history, strategies that insects and other organisms adopt. How many eggs do they lay? How much energy do they devote to migration in case of disaster or lack of food? How long do they live, etc. So we took into consideration all those factors. But some studies were done in the lab, some were done in the field. So what we did in the lab helped explain what we observed in the field. I hope that has explained. So it was not just about weather, it was about other factors, but we didn't quantify them. So we didn't sample for natural enemies, for example, because that was out of scope. And then about the biological species barrier, yes, this was the basic thing. First, are they biological species? And I'm happy, one of the students who worked on this part is here, because the proposal was presented in crop science. We didn't finish it on time. It's about what we call mating compatibility. So we tested these species, or whatever is suspected as cryptic species, are they compatible when it comes to mating? So we took that into consideration. And that's why we said in one of the species that we have evidence that they are not compatible when it comes to mating. And that's why we said these are different species. So we took into consideration that one. Then there is a question from Professor Wahabu Kimaro about control mechanisms, especially the use of weave ants. Now, I didn't show part of the slide that shows how we can use weave ants, but it's a very common question that people ask every day. And we always tell them, first of all, weave ants are very neat insects, so they don't like dust. So one of the easiest way of harvesting your fruits or tending your field is just to apply a little bit powder on your hands or some dust, and the weave ants will go away from you. They don't like it. Or we can apply artificial smoke before harvesting, just like we do with bees. If you want to harvest the honey, you can do the same technique. There are some protective gears to use when working with the weave ants. But also, when we do research in the field, we know the actual times when weaver ants are very active and when weaver ants are not very active. For example, if you harvest your fruits between 10 a.m. in the morning and 2 p.m., weaver ants are not very active. They're always retreating to their nests, etc. So there are so many different methods of what you can do, and we always give examples of how people harvest honey from the bees. So you can do the same. Phyllonoricta Mwatawala, this one is in the Mzumbe area. Unfortunately, the student whom we trained to work on these, is a Lepidoptera, the small Lepidopterans. The student whom we trained to work on these Lepidopterans, a bad thing about students is they graduate. So he graduated, which is very bad because you can't work now. So it's good, but it's bad. But we know where it is. We know how we sampled in the savannah area that Professor Tisekwa mentioned, where we have marula trees and some acacia trees. That's where we found the Phyllonoricta Mwatawala. Then a question from Professor Msogoya. He's asking whether we supported export trade, and that reminded me, when we started this work, we sampled banana and avocado. And we found that these flies attack avocado, which unfortunately was a very controversial host because avocado business is really big in Latin America. All those Puerto Ricans and Guatemalans, they like avocados, and it's a huge market for Latin America. And America was banning the export of avocado into the United States. So a lot of research was being done in Latin America area to prove that avocado is not a host of fruit flies. And what they realized is that one of the varieties of avocado, called Hass avocado, is actually an preferred host of avocado, of fruit flies. And because of that, the Americans and the American markets were opened. But along the same line, based on the data that we collected, unfortunately, we did not discriminate whether this is Hass or Ettinger or any other variety of avocado. But if you look at the incidences, they are very low, which indicates that probably we are sampling more of one variety which is not attacked, as well as some other varieties that were attacked. But in the end, what I can say is our export markets to South Africa for avocado were opened. So now we can export avocados to South Africa, but also we can export green bananas to United States. Because at first when we said banana was attacked, the market was closed. And when it was proved that the green bananas cannot be attacked, except the ripe ones with a split skin, then the markets were opened. So at least these were the export commodities that we know. Then you asked about the practicality of, I don't remember, using weavers. Yeah, it's very practical. I mean, if you look at the technology that we developed, it's very practical, and we want to make it more practical. That's why we want to rear them in the lab, so that someone can just get a nest from a screen house, rather than going out in the wild looking for the nest to introduce in the farm. So how far have we gone? I mean, we have gone from telling where they can fly and whether we can trap them, the queens I'm talking about, so we can trap the queen and we can rear them. And an interesting part is because these are social insects. It means if you move out of a colony and return, you are not their member, they kill you. So the queen coming from one colony, upon return, that queen has to go to another place. If that queen lands on the same colony, it's an alien, so they can kill it. And that's why we wanted to trap them. But it takes a lot of time for a queen to develop the caste, to have the workers, the males, all these. So what we tried was, can we try to make the queen accepted, or can the queen accept workers from other colony? And we tested that. So we put a small barrier and we introduce the workers, and slowly we think they will get used to this queen and the brood will grow quickly. So it's practical. And then a problem among small scale farmers. I mean, as I said, the most difficult part, the one that I showed, was about orchard sanitation. And this is something that our farmers do not like. It's a standard practice all over the world, whether it's United States of America, who are very advanced economically, or Tanzania. But our farmers, unfortunately, do not like this practice of orchard sanitation. And it's not just about mangoes. I mean, we have the kind of rules for cotton paste. You have to set timing that between these dates, you have to clean your farm, otherwise it's a criminal offense, something like that. We don't have that one for fruit flies. So maybe if we enforce it that way, we can suppress the population. If I'm given $1 billion, what would you do next? I'll go to Bahamas. Why should I worry about this when I have $100? Anyway, what I would invest more, based on these challenges, the challenge is to get a natural enemy, and this will also answer the question by Professor Tisekwa, is to get a magic natural enemy that will go and search for these flies in the wilderness, rather than spraying individual orchards. So we will increase more search for natural enemies, because these ones can do the work for us. Now we have some natural enemies. Apart from predators, we have a group of natural enemies called parasitoids. But their parasitism level in the lab is very high, but it is very low in the field. So like 7%. So we would be very happy if we can get up to 90%. So that's the area I would like to invest, because then they will do the work for us. About marula. Yes, marula are everywhere. I mean, we have them behind animal science, and some of them, that area near Mzumbe, Melela area. But as I said, the only way to control fruit flies in marula would be to get such kind of parasitoids that can go actually and search for the fruit flies in the wild. And we have some that have been introduced. As I said, they're not doing very well. So we have 2 parasitoids that have been introduced, but so far we have never been very successful in that area. House flies versus fruit flies. So far, fruit flies have never been proven to be either active or passive vector of any disease. Then there are no any records that eating an infested fruit full of maggots will cause you any harm anyway. So my friends from Zanzibar, they say that nakula embe gizani, so you don't worry about the maggots you eat. So You're not going to suffer anyway, sure. So there is no problem. We just fear the maggots. So Dr. Madala asked about pests. I mean, we are regarding these as pests, and he would like to see them as a source of protein. Yes, we capture a lot of fruit flies in the traps, but we were thinking of using them as chicken feed. We didn't think about your fish, unfortunately. So we'll start thinking that way. But also, when we are working with weaver ants, the queen of weaver ants is big, and it's a delicacy in Asia. So one of the things that we wanted to test in this program was whether we can harvest those queens of weaver ants for sale in countries like Thailand and Vietnam. Unfortunately, we could not get a huge population of queen, and that's why we were developing different methods of rearing the queens in the nursery so that we can harvest them and sell them abroad, because it's a delicacy in Thailand, Vietnam, China, and other parts of Asia. Now, Dr. Mbije is not from Department of Crop Science and Horticulture, but Helen Mbije is, so Mrs. Mbije is from our department, so that's fortunate. The Association of Mango Growers, these people who, it's like a lobbying group for mangoes, they would like to see 3 varieties as their flagships, the flagship varieties. So, as you're saying, there are so many varieties, but for them, they would like to be confined to a few varieties, and those specialists in fruits will help me. They would like to sell 3, apple, mango, Kent, and Kate. 3 are their flagship varieties. But we, the ones who buy them, and that's why you should come and buy your mangoes from us, from Crop Science, not on the streets. You just want the red mangoes of Sua, and they give you everything. It's just red. This is red. Tommy, Kent, Red Indian, whatever that is red, they give you. And later on, you realize you have a mix-up of varieties in your farm. And that it's been going on that way for so long. So we have that one as a source where you have so many people selling everything because these are red. But also we have so many uncontrolled imports. I think, I stand to be corrected, because we have mango that they call them mango from Mombasa, Embes are Mombasa. And they normally come during the off-season of our main season here. So whether they are subject to quarantine and cross-border processes, I'm not sure. But those are the causes. But consumers have their preferences. How my friend Dr. Kasui says he doesn't eat these mangoes, he only eats boribo from Tabora, his hometown. So, thank you so much.
Colleagues, it is almost time, but I'm flexible. Should we go for the second round? One more talk with sentence.
Thank you, Mr. Chairman. I have only one question. First of all, I would like to congratulate Professor Mwatawala for a nice lecture. But my question is on the host. You have talked a lot about the variability which is in the fruit fly species. But my interest as a plant breeder is on the variation in the host plants. And we know that we have different types of mangoes, like you said now, that there are different types of mango. In your work, in your research, did you see that there is any variation in the fruit fly attack on these different types of mangoes? And also, did you see any kind of fruits that are not attacked by the bean fly? Not bean fly, because we have another fly, by the fruit fly. I'm a bean person. I'm saying this because if we find a kind of fruit that is not touched by a fruit fly, that could be a source of resistance, which you could use using even genetic engineering to transfer those genes of resistance in edible fruits. So did you see any kind of variation in any fruits? Thank you so much.
Thank you very much, Professor Mwatawala. Big up for a nice presentation. I'm congratulating you for sitting in so many committees and boards, TFDA, TPRI, PQPS, etc. But the great challenge I'm seeing is the introduction of these new invasive species of different pests into our country. And you are working closely with the Ministry of Agriculture as well. What are the strategies which you are putting forward to protect our country? You can see now we have fruit flies, we have fruit absoluta, we have now fall armyworm. You can mention a lot of new invasive pests in our country. And in the long run, we cannot even go to these international trades unless we have a very stringent measures. Can you give us a suggestion? Thank you.
Last one.
Professor Mwatawala, hongera sana. I used to sit with Professor Mwatawala somewhere. I'm glad for your excellent presentation, and I'm happy that I have acquired another new concept of PRA. I thought PRA stands for Participatory Appraisal, but we know now it is Pest Risk Analysis. So thanks for that new addition of the concept. So my question is, your presentation on facilitating international trade through science, with that case of fruit fly, what theory have guided your presentation? Thank you.
Last one.
Thank you, Mr. Chair, and let me congratulate Mwatawala for a wonderful, wonderful lecture. My name is Mark Mamu. I know at the beginning the chair was making reference to students in the audience. This is probably yesteryear students too. I'm a veterinarian. I was very attracted to this lecture, and I must say I was very, very pleased that I did come to this lecture. What attracts me is systems. And maybe I want to ask you a question now in your elevated position now in so many committees and now distinguished professor. So evidently you've had plenty of time to think through some of these issues. I noticed one of your long-term collaborator is called Mark. So I can take the liberty of replacing Mark in this room with this Mark. What you described about interactions with the people in FAO and the international, these plant pest issues. And at the time in FAO we were developing a program called Empress. Later on we were looking at long-term impacts of risk of infectious diseases. We're looking at humans, animals, and plants. So I got interested in these, what commonalities. Now, much of what you've described evolves around the concept of detection, identification, monitoring with a view to risk management. That's the term which we use here. But you just condense that into two. Why detection and identification, and also probability of risk. Those are similar concepts. Now, in a university like ours, it cuts across sectors. When you have these underlying concepts, to what extent can we get certain concepts which SUA becomes world leader in, or African leader in? The second question comes into that is, from your new position, I got to know about this pest, which was inspected by a UN agency, not FAO, by the way, Scania. There was a litany of new pests coming in, and so on. So that brings the issue of early detection, very good. But how do you assess now our efficiency for early detection, for quarantine and response, and preparedness? Or are we drifting into a situation of fatalistic situation, notwithstanding all these kind of floating signs? Sama. I hope Vice Chancellor will excuse me if I've on inter-college issues.
Thank you, Mr. Chairman. Let me join others in congratulating Professor Mwatawala. I'm congratulating him from the best part of my heart for the presentation. And the chair just received my apology for having come in a bit late. But I have one question. Professor Mwatawala, you have really done a very wonderful work, and I think which will elevate our university from where we are to the next level. But I want to know one thing. This work you have done, it has a big potential in assisting our country in controlling some of these new and dangerous pests. To what extent have you, engaged with the ministry responsible for agriculture to make sure that whatever will really, at the end of the day, come out from your body work will be internalized into our policies, and at the end of the day, be advocated by the responsible organ so that it can be put into use. Thank you, Chair.
Okay, thank you so much. So I'll start with the question from Professor Nchimbi on the variability of the hosts. And I might say that from the mango varieties that we tested, Tommy, Kent, Kate, Red Indian, and some local varieties like Sindano Nyeusi, when we did the statistics, we didn't find any significant variations in incidences or infestation rates. But if you walk around, people will tell you your new mango, like we are the one who brought them, your new mangoes are more infested and the old mangoes are not infested. So I don't know where that hypothesis come from, but it's a very common hypothesis that the improved varieties are attacked while the local varieties are not attacked. But no research, no right to speak. We have data that can prove they're attacked in the same way. The other examples I gave was on avocado. As I said, you have one variety which is Hass avocado, which is not preferred host, while the others like Ettinger are probably preferred host. Now, one of the theories that came out, I don't know whether it works or not, but they said that Hass avocado has some compound in its skin that can encapsulate the egg of the fruit fly. So once the female oviposit an egg, it is quickly encapsulated by some chemicals, and it cannot grow any further. So maybe that one will help. But among the hosts that we sampled a lot, and they couldn't bring anything, is the star fruit, Calambola. So, but that one has a lot of water, so rearing of the fly from that fruit was not positive. But from other country, it is reported that star fruit is also positive for fruit flies. Otherwise, from our sampling also, papaya was not a positive host. So we did not rear any fruit fly, although it's a known host in other countries, but from our samplings here, it's not known as a host. But then there are so many different varieties which we didn't take into account because we didn't plan our studies to sample by varieties. It was just by a fruit species. So I wouldn't say much about host plant resistance because it has not been very successful in fruit flies control. A question by Professor Mamiro is that how would we move forward? Suggestion on how do we move forward? And it's always a difficult question because of our position, because as researchers, even though people say we run away from the problem, that as researchers, we generate information. I mean, that's the main output of research, is to generate new knowledge. That's our objectives are to determine, to establish. We are not developing, I mean, like Professor Makundi said. Because development now takes into account so many other factors, interests, values, and so many other issues. So as an individual, I can't say anything, what I can do, but as a person who sits in those committees and boards that you said, we always try to influence them. But Dr. Mamiro, you know, I mean, Professor Mamiro, you know, because we sit in some of these committees, there are so many issues when it comes to invasions. I'm talking about Bactocera dorsalis that we detected in 2003, but it is yet to be reported today. So officially, it's not in Tanzania. So all those pests that you mentioned, Tuta absoluta, fall armyworm, officially, they are not in Tanzania. So I will end it that way. Babili Innocent, my brother from 5 Science 2, Mazengo, so we say, come a long way. He is asking about theories. Now, I'm not very prepared to answer that question because the social scientists have so many complications when designing their study. We don't do that. We only go for knowledge gaps. If you ask me what were the gaps before, I will tell you we didn't know 1, 2, 3, 4, 5. But telling you that if this one rises, this will go down, that one will run away. Now, unfortunately, we are not guided that way. And I have another brother here who inspired me a lot, and he is like one of those people who raised me. He's an engineer, but he decided to do social sciences. So engineer Issa Majid is with us today, and he's always coming to me asking me, why these social scientists? And I tell him, yes, you decided to join them, so go along their lines. So thank you so much, my brother. There is a question from Professor Mark, but that one I'll keep it the last because it was a bit long. I'll go first to Professor Chibunda's question. That's how much have we engaged with the Ministry of Agriculture? So we have engaged a lot with the Ministry of Agriculture. Of course, there is always some these entities reluctance, but in the end, we managed to work with them, and we developed that management plan because, first of all, we wanted to develop a policy document or something. And the ministry people told us, you are an institution, you cannot develop policies. Give us a management plan and we try to adopt it into our agenda. So that's what we did. So we sit together with the people from Ministry of Agriculture and came out with the management plan. And on the same year, it entered into their records because it became part of the speech of the Minister of Agriculture, then not Tizeba, the other one, Chiza. So it was mentioned in the parliament, so I think it's part of the answer. So at least if they want to do anything about fruit flies, they will always refer to the management plan. But along the same line, we printed, out of that project, we printed so many, close to 6,000 or 10,000 copies of a Swahili version of the management manual. And we made sure that all these councils where they grow fruits received quite a good number of those booklets. And then the Minister of Agriculture formed what they call team of experts, and they co-opted us. So we sit in their team of experts for fruit flies. And then we sit in their plant quarantine subcommittee together with Professor Mamiro, where we also engage very well on these quarantine issues. But Professor Sibuga, who was boss of the our section on crop protection, she's the chairperson of the National Plant Protection Advisory Committee, and our subcommittee reports to her. Now, the other question from Professor Remam, yes, the other person is Mark, but is a Mark with a C. I think yours is with K. So we can switch. Yes, we go along the same line. I mean, the whole concepts, we look closely at your projects. So the surveillance and detection, as you said, they go along the same way. I mean, you deal with animals, we deal with plants, but sometimes the techniques are the same. So one day I know I was speaking to one of the students of Professor Misinzo, and I was talking about these cryptic species. And the student was like, so you are doing the same stuff like us? I said, they're just species. I mean, if your viruses are cryptic, I don't know how they look at viruses, but if they're cryptic, just like at fruit flies, we can go molecular, we can go to cytogenetics, we can do all these kind of studies. So if we can come up with a center for detection and surveillance, which we do not have for the whole of SADC region, and that will be great for both you, infectious diseases as well as plant diseases. The surveillance system for our ministries, most of them in this region, is not existent. And that's why you have all these, invasions. And it's easy to test. I mean, just bring a mango from India and you are safe with it into Tanzania. No one will bother you unless you are very unlucky. But I'm not telling people to smuggle fruits. But our surveillance system is not very well established. I cannot say it's not working. And it's not just for Tanzania, but for other countries. And I went to I explained this in one of the meetings, and these people, because they're not from this region, where they wondered, how can you have people crossing borders, I mean, without anything, without ID, without anything. And I joked to them that there used to be a house in Mbeya, where I come from, in Tunduma, where a sitting room was in Tanzania, and the bedroom was in Zambia, because there was no clear border. So I cannot tell people to not bring the fruit to the bedroom because it's in Zambia. So our borders are very porous, etc. So if we can have one coordinated regional surveillance system, because I tell people, a Masai from Kajiado, we cannot tell him that do not go to Longido, because probably the other families in Longido. And so we are so much interconnected across borders, and we cannot control it unless you have a regional surveillance program. And that will be for both, because the cattle keepers, they carry fruits with them, etc. And then early detection. I mean, this one is not whether efficient or prepared. It's simply non-existent, because even I hear there used to be traps at SUA for detecting things like armyworms, locusts. We don't have them anymore. So the whole detection system, surveillance system, need revamping. It's not existent. The only people we know who work on, I mean, as far as crops are concerned, is the center in Arusha. And they are more concentrated on locusts and armyworm. The original armyworm, not the fall armyworm. So if I can answer your question in Russia, because it was a bit long, that we need a regional center, coordinated center for surveillance, detection, identification. I mean, same words as you said, how you use them and how we use them. And then if we can revamp now the surveillance system, because according to FAO, we cannot do it. That's why I detect latifrons, but I have to tell the Minister of Agriculture to announce whether Tanzania, I cannot do it. Legally, it's not possible. So they need their surveillance system activated, and then we can feed them with the information, and they can take up the legislative and policy issues ahead. Thank you so much.
Ladies and gentlemen, I think, as I said in the beginning, you will agree with me that this lecture was very educative, was very clear, and as I warned in the beginning, there were going to be so many questions. And counting the number, there were more than 15 questions. Thank you very much for asking questions, because really it showed that you were interested in the lecture. And also Professor Mwatawala, thank you very much for responding to various questions which were put forward. Ladies and gentlemen, through this inaugural professorial lecture, we've been told that much has been done on fruit flies. So with these few remarks, I thank you again, and I also thank the management for appointing me to chair this very important inaugural lecture, and also for turning up and staying all this time up to now. May I now declare the Mwatawala's Professorial Inaugural lecture officially closed. Good night.
Naitwa Profesa Maulid Mwatawala na kwa sasa ni mkuu wa idara ya sayansi ya mimea na kilimo cha bustani, na ndo nimetoa hii inaugural lecture inahusiana na jinsi gani sayansi inaweza ikasaidia utekelezaji wa biashara ya kimataifa, na tulikuwa tukiangalia zaidi jinsi gani biashara ya matunda inaweza ikaboreshwa kutokana na utafiti wa waharibifu wa matunda na zaidi na onzi waharibifu wa matunda. Mimi nawashauri tu, sababu na wadogo zangu wengi tunafanya nao kazi, kwa hiyo mimi nawashauri tu kwamba wajipange hivyo. Yani mimi niliwaza hivi miaka 9 iliyopita. Ilikuwa siri yangu, sikumwambia mtu. Lakini nilitoka hapa IC na nilikuwa nimesimama hapa, nikamwangalia Profesa Makunda anachokifanya na mimi nikaona kwamba kuna siku nitafanya. So ilikuwa ni miaka 9, wakati usijawa pengine hata profesa, labda nilikuwa senior lecturer. Lakini nilikuwa nishaanza kujipanga, na ndio maana hata story nilikuwa naitoa, unaona ilikuwa inaenda kwa chronology, kwa sababu nilikuwa najua kwamba labda 2013 nitakuwa nimemaliza research hii, 2014 nitakuwa na management, 2010 na ngapi story yangu itakuwa imekamilika. Kwa hiyo japo haitaisha on time, lakini angalau unakuwa na hiyo plan.